Hello, my name is Yesid, and today I'm going to talk about the protein digestion in monogastric animals. Monogastric animals are characterized by having a single-chambered stomach, unlike ruminants with a multiple stomach compartments. Some examples of the monogastric are humans, pigs, dogs, and poultry. So most monogastrics are generally unable to digest much cellulose food material such as grasses, but herbivores with a monogastric digestion system like horses or rabbits, they are able to digest cellulose in their diets through microbes in their gut, but they extract less energy from these foods than do ruminants. Well, before to start to talk about the digestion, I would like to talk a little bit about proteins. So proteins are macromolecules composed of amino acids linked by peptide bonds. They are involved in the structure, function, and regulation of cells and tissues. So the proteins are the mainly components are amino acids. So these ones are like building blocks of proteins. There are 20 standard amino acids, each with a unique side chain. There are some essential amino acids that must be obtained from diet, and the other ones are non-essential amino acids that can be synthesized by the body. Proteins have four structural levels. So primary, secondary, tertiary, and quaternary. So the specific arrangement of amino acids determines the protein function. So in this figure, you can see the example of how the protein are made. So the primary structure is a sequence of amino acids in a polypeptide chain. The next level of protein structure is the secondary structure that refers to the local folded structures that form within a polypeptide due to interactions between atoms of the backbone. And then the overall three-dimensional structure of polypeptide is called a tertiary structure. So the tertiary structure is primarily due to interactions between the air groups of the amino acids that make up the protein. And finally, the quaternary structure is the multiple polypeptide chains that are also known as subunits which form the completely protein. So this makes the real protein structure. So this is the protein that we are going to see how is the digestion process in the monogastric animals. Well, now with this brief introduction about proteins, I will start talking about the monogastric digestive system. So the first part is the ingestion of feed begins in mouth. They start to chewing, and the production of saliva both help with digestion. So chewing breaks down food into smaller components and increases the surface area available for enzymatic digestion. In some species, saliva contains salivary amylase, that is an enzyme that will work for break down the starch. Then start the next portion of the digestive system is the esophagus, which connects the mouth to the stomach. So in this section, the transport of the food that were chewed to the stomach through the peristaltic movements. Then the next portion is the stomach, which is a muscular organ with distinct regions, like generally our cardiac region, fundus, body, and pylorus. So in the stomach, you will find gastric glands that secrete gastric juice, which contains hydrochloric acid, and enzymes like pepsin. So the gastric acid and pepsin facilitate the breakdown of proteins into smaller peptides. The stomach pH is highly acidic, so generally ranging from 1.5 to 3.5. So the low pH is primarily due to the secretion of hydrochloric acid by the parietal cells in the gastric mucosa. Well, now it's important to mention that protein digestion in monogastrics, so this starts in the stomach with the pepsin breaking down proteins into peptides. Then continue in the small intestine with trypsin and chymotrypsin converting peptides into amino acids. However, I will talk a little bit more about this digestion process in the next slides. So the basic thing that occurs here in the stomach is denaturation, which is a process in which proteins lose their structure, typically their secondary, tertiary, and quaternary structure, without breaking the peptide bonds. So the stomach acid helps to denature proteins, unfolding their three-dimensional structure, so this makes the protein more accessible to digestive enzymes. In this case, the pH also has some influence because the acidic pH of the stomach facilitates the protein denaturation. The low pH disrupts the hydrogen bonds and electrostatic interactions that maintain protein structure. So I was mentioned before, the acid and pepsin break down proteins into polypeptides, but we need to understand that these enzymes are not activated until there is a mechanistic process that helps to activate the enzymes to help to this breakdown proteins. Well, in this figure, we can see a little bit about the denaturation of the protein. So, because the acidity of the stomach causes food proteins to denature, so this means that there is occurring an unfolding their three-dimensional structure to reveal just the polypeptide chain. So this is the first step of the chemical digestion of proteins, as I was mentioned in the previous slides. So remember that the three-dimensional structure of a protein is essential to the protein function. So the denaturation in the stomach also destroys the protein function. So in this figure you can see the more or less what is the quaternary structure. Then because the hydrochloric acid function destroys a little bit the structure, and you can see then the different colors of which means that are the peptides. However, it's important to mention that there are some factors affecting denaturation. One of them are the temperature, because the higher temperatures can accelerate the protein denaturation. So the combination of elevated temperature and low pH in the stomach can improve this process. Also the time, so the longer exposure to the acidic environment increase the extent of denaturation. And the last one is the concentration of the denaturant. So in this context, is the denaturant is the low pH in the stomach. So higher concentration of hydrochloric acid can contribute to more extensive protein denaturation. Well, when the enzymes break down the protein in peptides, then these continue to the small intestine that works similarly across species despite dietary difference. The main function of the small intestine is to move nutrients from the diet into the bloodstream. In a herbivore, this includes the non-cellulose part of the diet, fats, protein, carbohydrates from grains or oils, rather than hay. Once the feedstuff enter the small intestine, they are mixed with enzymes and buffers from profuse pancreatic secretions and from the small intestinal brush border. It's important to mention that liver and pancreas have some function for the protein digestion. Well, specifically the pancreas, because the pancreas secretes digestive enzymes and bicarbonate to neutralize stomach acid. So the pancreatic enzymes are released in an inactive form that are called chymogen, and these enzymes must be activated to start the digestion process. Otherwise, the enzymes can be affect the cells that are in the intestine. So this is an important section, because the small intestine is very important because the majority of amino acid absorption occurs in this section. So particularly in the jejunum and ileum, this is the final step in the process of protein digestion, which begins in the stomach with denaturation, and as I mentioned before, and continues with enzymatic breakdown in the small intestine. So here, like I mentioned before, the pancreatic enzymes like trypsin, chymotrypsin, lipase, and bile from the liver aid in further digestion. So, well, now that I already explained how's the process, starting in the mouth, go to the esophagus, move then to the stomach, and then the products, the peptides, go to the small intestine. So I will mention now some proteolytic enzymes that are involved in all of this process. So the pepsin is secreted by gastric chief cells in the stomach. It functions in an acidic environment and cleaves protein into smaller peptides. Then you will find the trypsin, which is produced by pancreas and is activated in the small intestine. The trypsin cleaves peptide bonds on the carboxyl side of basic amino acids. The chymotrypsin also produced by the pancreas and is activated in the small intestine. The chymotrypsin cleaves peptide bonds on the carboxyl side of aromatic amino acids. And there is other one that is called carboxypeptidase, which removes the individual amino acids from the carboxyl end of a polypeptide chain. So as I mentioned before, the proteolytics enzymes are not ready to use. They need to be activated because if they are activated all the time, they will make some problems in the intestine cells. So many proteolytic enzymes are synthesized as inactive zymogens or proenzymes to prevent self-digestion. So the activation often involves the removal of a specific peptide or protein fragment to expose the active site. For example, trypsinogen is activated to trypsin by enteropeptidase in the small intestine. I will talk a little bit more about how the activation of some more of the mainly enzymes in the next slides. So in this figure, you can see like the hydrochloric acid creates an acidic medium inside the stomach, which helps in the activation of pepsinogen to pepsin. And also the active enzyme pepsin is responsible for breaking down proteins into proteases and peptides. In the case of trypsinogen, the trypsinogen is an inactive form of trypsin. When trypsin is activated by the enterokinase enzyme, which is secreted by the cells present in the intestinal mucosa, the trypsin also helps in breaking down protein into small peptides. So trypsin works better under alkaline pH of 8. So it is activated by enterokinase in the small intestine and in turn activates other pancreatic enzymes like chymotrypsinogen, proelastase, procarboxypeptidase, and prolipase. Well, after the peptides are digested for the enzymes in the small intestine, then there is other part that is large intestine, which includes the cecum, colon, and rectum. So in this portion, there is water absorption and fermentation of indigestible carbohydrates occur here. There is a few digestion and absorption of the amino acids. There are not too many receptors in this portion of the digestive system. So as a summary of the enzymes, I would like to mention the enzyme, the substrate, and the end product. So the first one is the trypsin. So the substrate are the polypeptides from the stomach, particularly those containing basic amino acids. The end products are smaller peptides with basic amino acids. The next enzyme is chymotrypsin. The substrate are polypeptides from the stomach, especially those containing aromatic amino acids. The end products are smaller peptides with aromatic amino acids. The carboxypeptidase is an enzyme that is present in the small intestine, and the substrate are peptides. The end products are amino acids. Aminopeptidases, the substrates are peptides and the end products are amino acids. There are also dipeptidases, the substrate are dipeptides and end products are individual amino acids. And then there is the enterokinase that is present in the small intestine, and substrate is inactive trypsinogen. The end product is the activated trypsin. So as you can see, the proteolytic enzymes have different targets, so it depends on the characteristic of the amino acids in the peptides. So for example, some enzymes the target are basic amino acids. So there are three amino acids that have a basic side chain at neutral pH, which is arginine, lysine, and histidine, and other pancreatic enzymes. The target is the aromatic amino acid. So in this case, is the amino acids that includes aromatic ring like phenylalanine, tryptophan, and tyrosine. So as you can see here, there is a table with the different characteristics of amino acids. So the enzymes will be make a cleavage directly to the side of where the specific amino acid are for target of this enzyme. So after the talk about the proteolytic enzymes, there is also a regulation of these proteolytic enzymes that I would like to mention in this presentation. So there is the feedback inhibition. The end products of protein digestion can inhibit the activity of the proteolytic enzymes to regulate the overall process. And also the pH sensitivity. So many proteolytic enzymes have the optimal pH range for activity. So, for example, pepsin functions optimally in the acidic environment of the stomach, a difference than the other enzymes. Well, now let's move to the mechanism of amino acid absorption. So, active transport is the first one, which is amino acids are primarily absorbed through active transport mechanism. There are some specialized carriers known as amino acid transporters that facilitate the movement of amino acid across the epithelial cell membrane. And the other is the co-transport with sodium. So many amino acids transporters use a co-transport mechanism with sodium ions. This process is energy dependent and is crucial for maintaining the concentration gradient required for efficient absorption. So in this figure, you can see like a little bit some explanation of how works the absorption of amino acids. So amino acids are absorbed from the intestinal lumen by a group of transporters on the apical surface of enterocytes that couple their transport to the transport of sodium ions. At least there are like 7 different carrier proteins that transport different groups of amino acids across the enterocyte plasma membrane. So in this figure, we can see the co-transport of sodium from the outside of the apical membrane to the inside of the cell is driven by the low intracellular sodium concentration. The low intracellular concentration of sodium is maintained by sodium-potassium ATPase on the basal-lateral membrane, which actively transport the sodium out of the cell in exchange for potassium. This transport maintains a sodium gradient, which allows the cells to concentrate amino acids from the intestinal lumen. So the amino acids leave the enterocyte by a facilitative transporter on the basal lateral surface of the enterocyte for the entry into the bloodstream. So depending on the molecule, there are different receptors. So for peptides, in the apical site, there are coupled transport of hydrogen ions, and for the basolateral, they are independent of hydrogen ions. Well, for the amino acids, there is the apical uptake. There are symporters and antiporter transporters. It depends if they are anionic, they are acidic, or if they are cationic, they are transporting basic amino acids. And also there are the basolateral efflux, which use the antiporter transporters. So once the amino acids are in the blood, they are transported to the liver as with other macronutrients. The liver is the checkpoint for amino acid distribution and any further breakdown of amino acids, which is very minimal. So dietary amino acids then become part of the body's amino acid pool. And then assuming that the body has enough glucose and other source of energy, those amino acids will be used in one of the following ways. They can be made for protein synthesis in cells around the body, making non-essential amino acids needed for protein synthesis, making other nitrogen-containing compounds, or if the amino acids that are not absorbed in the small intestine, they maybe can can be absorbed a little bit in the colon, but if they are not absorbed finally, they will be excreted in the feces. Well, thank you so much for watching my presentation. If you have any comments or questions, please let me know. Goodbye.